Okay, welcome back to Biology 103. We're ready to begin Unit 3. In Unit 3, we will be talking about mitosis, also meiosis, and a little on birth control. We may get to a little bit on development. Okay, so a karyotype. What you see before you is a map basically of all the chromosomes, showing you the size of them, showing you the shape of them, and this is showing you maternal and paternal chromosomes that have been passed down to this person. Shows you also the sex of the individual. So if you can take a look at this karyotype and tell me, number one, what, if anything, special that you see, and then also try to sex the individuals. So the one at the top, we could say is A. The one at the bottom, we could say is B. So somebody tell me, is A boy or girl? And what did you come up with? Well, if you said girl, you're correct. Why? Because this individual has 2 X chromosomes, so we know that this is a female. And then the one at the bottom, if you said it was a boy, you're correct because he has 1 X and 1 Y chromosome. And look at the size of them. The X chromosome is much bigger than the Y chromosome. But yes, this is a male. My pen stopped working. Let's get over here. A male. Alright, so it says, what is a karyotype? Again, it's the appearance and characteristics of the chromosomes that are found inside of a cell. Things that it shows us, especially the size, it shows us the shape of them, and it shows us the number. Okay? We said one of the other things that we were saying is that one of the other things that we want to say about the chromosomes, all of the chromosomes, okay, down through 22 is what we call autosomes. These are the chromosomes that carry on all of the bodily functions, for example. But then there's some chromosomes we said that not only carry on some of those other functions, but they do other things such as helping to determine your sex. So the sex chromosomes, we said an X, and an X makes a girl. We said an X and a Y is going to make a boy. Okay, so let's go ahead. Chromosome structure. We're going to talk about the cell cycle, but we're going to talk about this a little bit before. This generally happens in prophase, as far as like the condensing of the chromatin net into a chromosome, the replication of the DNA that basically occurs during the S phase, during interphase. Okay, so in interphase, that's when you're going to synthesize and make a copy of the DNA. So what we have is when you have 2 identical copies of DNA, because when you're going through mitosis, you need to be able to pass an identical copy of that chromosome material on to the new cell. So, and an easy way to pack it so we don't lose any parts is in a chromosome structure. So what you see are 2 identical copies of the chromosome material. All right, so let's talk about this structure specifically. The arrows at the top, they're pointing to the 2 identical chromosomes, so we call those sister chromatids. The yellow arrow is pointing at the region where we hold them together, which is called the centromere region of the chromosome. So we say it again, chromatin net condenses 2 chromosomes during prophase. We said the 2 identical copies are called sister chromatids. Remember, we said the red arrows are pointing at the 2 identical copies of the chromosome. So we have a double helix and another double helix, okay, of DNA, so that each new cell has a copy of that original DNA. And then we said they are held together at the yellow arrow, which is called the centromere region. So the centromere region holds those 2 copies together. All right, so let's talk about why cells reproduce. If we're talking about a unicellular organism, we know that that unicellular organism is going to reproduce in order to create the next generation, okay? There's only one cell, so we know that that's why he's doing it. But a multicellular organism is going to make copies of his cells, and we're not talking about sex cells, we're talking about the body cells, okay? So the multicellular organism is going to need to repair. You probably bumped in. So say, for instance, for us, we've got cut, we probably got sunburn, we probably bumped into a desk or something, had a bruise or something internally that we didn't even see. We're constantly shaving, cutting stuff off. So it says we need to repair and replace that damaged tissue. Plus, at one point in time, you were just one cell where the mother and the dad's DNA united inside of the egg or ovum, and then begin to double the cells, and then take those two cells and replicate again, and then you've got 4 cells, so on, so another for growth, okay? And so we said for repair, growth, and then you developed into this multicellular organism whose body can carry on many different functions. So development. So for repair, growth, and development. Okay. Now cell types. Somatic cells. Again, we said those are adult stem cells, non-specialized cells whose job is to divide and produce in all the different types of cells in the body. And then we also have germ cells, which we're referring to as sex cells. Sex cells, gametes, they are going to make a new you. Okay, so again, we have some cells like muscle cells, bone cells, okay, cells in the dermis, epithelial cells, okay, that are inside of a blood vessel. All these things carry on the daily function. Sex cells, they determine or make a new you. Okay, now the cell cycle. It says in order for us to make a new cell, then this cell is going to have to go through what is called the cell cycle. Now, when a cell goes through the cell cycle, you'll notice that the cell cycle is divided into multiple parts. Okay, it takes anywhere from 8 to 20 hours to replicate a eukaryotic cell. Okay, so a cell division is referred to as the period between 2 cell divisions. So in other words, when the cell divides, okay, and goes all the way around this cell cycle until it divides again. So that is the cell cycle. So the period between 2 cell divisions. All right, the cycle is divided, you can see, into 2 main steps or 2 main divisions. The 2 main divisions are what you see here, which is interphase, okay, which is where the cell spends most of its time. And then you also see what? Then you see in between here, mitosis. So mitosis. Okay, you'll see that interphase has 3 periods. You see G1, S, and G2. Again, those 3 periods are G1, S, and G2. And then you see mitosis. Now, mitosis is actually divided into 5 different phases, and you'll see prophase, prometaphase, metaphase, anaphase, and then telophase. Okay? Now, one of the things, if you remember, Interphase was formerly called a resting stage. This is before they knew any of these events that we're going to talk about a little later were happening. They thought the cell was at rest, and then all of a sudden, then this cell goes into this event that we call mitosis. But in fact, the cell was not at rest. We know that G is standing for growth. So G stands for growth. So the cell is growing. So that S stands for synthesis of the DNA. So in other words, we're making things and going through cellular respiration and all this other kind of stuff in order to get this cell ready to divide and to be able to provide all the necessary components to have a new copy and a functioning cell. All right, so now one of the things that we want to look at is the fact that the cell cycle has chemical signals at the end of interphase that trigger mitosis. So not only are those triggers or signals at the end of interphase, but they're throughout the cell cycle. But we want to make sure that this cell is ready before we actually enter into mitosis. Okay. All right. So let's look at where these checkpoints might be. And this sounds like an excellent essay question to appear on your test. All right. So again, these different checkpoints, they're either going to inhibit or stimulate the next metabolic event. All right. So we said we're going to find checkpoints at G1, checkpoints at G2, and checkpoints at M. And so what we want to do is examine what's going on at each of those checkpoints. Okay? Because the whole idea for these checkpoints is that they allow time for any damage that might have occurred, any mutations in the DNA that might have occurred that we don't want to pass along to a new cell. So let's examine these checkpoints. Okay, so all right, so we said we wanted to examine the checkpoints of the cell cycle. We said the first checkpoint. So first, let me try to draw the cell cycle one more time. We said G1, S, and G2. Now, if you remember from the last slide, it said that this is where we get our first stop or checkpoint. So the G1 checkpoint. Now remember, checkpoints are different from the stages that are in interphase, even though they may include some of the same letters, but it's different. So we're talking about the checkpoints. Okay, so the G1 checkpoint, it says you've probably had something to drink. So that means that you've been exposed to whatever contaminants that were in whatever you drank, whether it was water, whether it was some alcohol, whether it was a Coke or whatever. You've been exposed to that. You've also been exposed to other environmental factors. You've been exposed to the sun. So in other words, you've breathed some things. It says all of these things can affect our cells, and more specifically, they can affect our DNA. You may have just sprayed for bugs in your house, and then you didn't have any shoes on, and you're walking through the pesticides. Nobody likes spiders, so I can't stand spiders. And at one point in time, I had just spiders just popping out, hanging in front of my face. So you need to, if you're afraid of spiders, you need to spray for them spiders.